much, Robert, and thanks very much. Let me just wait a few seconds while we got chaos.
Yeah, thanks.
Okay, so thanks, Robert, and thanks very much to the speakers that came before me because, yeah, it's already informed a little bit maybe of what I'm going to say, so that's excellent. Right, so I'm going to be talking about like sort of the last 10 years of work that we've been doing on angling selectivity and trying to discuss how it will potentially influence the vulnerability of capture to fish. And so what we do know is there's no doubt, and you've seen it in the previous talks, that fishing with hook and line does selectively remove certain individuals. It reduces diversity in fish populations, and it certainly selectively removes individuals with fast growth rates, and it selectively removes bold individuals, and we know that from the work that Robert's done, Robert and co. But what are the actual mechanisms behind that? And that is some of the things that we've been discussing here. Is it learning behavior, cognitive behavior? What is it that's driving it? And we really wanted to test whether it was metabolic physiology, so aerobic physiology, that had an influence on these things. And the reason for that is because we know that there's a link between growth and physiology of fishes. We know that there are links between boldness and physiology of fishes. And one of the methods driving reduced growth and boldness may, in fact, be the selective removal of certain physiological phenotypes. And so we wanted to test whether this hypothesis might be true using a resident reef fish that we had along our coast. So the first thing that we did was we went and looked at a population level, and we looked at two populations, one inside a marine protected area, one outside a marine protected area, but this was population level. So we looked at individuals over a temperature range. One individual would be at one temperature, and so in so doing we built up a population. And what we did see, that at thermal extremes, there was definitely a significant difference in the aerobic scope at a population level. And we speculated that exploitation might actually be removing higher performance, physiological performance phenotypes from the fish population. But in order to test that, you can't work at a population level, and you have to work at the level of individuals. So we went on and we did a very similar thing from an exploited area and a comparable unexploited area, and we looked at individual aerobic scope curves. And when put together, and then we also tried to classify those performances into low, intermediate, and high physiological performance based on their performance across a thermal range. And when you plotted it all together at the population level, you could see that the same patterns were emerging, where the exploited population outperformed, the unexploited population outperformed the exploited population, particularly at thermal extremes. But perhaps the most interesting part was that there were no high physiological performers in the exploited population, which really showed us that these high physiological performances were being selectively removed from the fish population. We wanted to then see if that was reflected in the wild in terms of activity. Because if we understand activity, we know activity is a critical component of whether a fish is vulnerable or not. The more active it is, the more likely it is to encounter a gear. And so highly active fish are more likely to get caught, in theory. But we had a look to see if the aerobic scope matched the activity on the water, and you can see the aerobic where's the pointer? Oh, there we go. You can see this is the aerobic scope curve. How do I go back? Oh, there we go. And below that, here's the activity measured as acceleration. We used accelerometery telemetry, and you can see a similar pattern emerged with higher activity at thermal extremes in the unexploited population. And similarly, in another MPA, we had with the similar patterns, with definitely, in this case, higher activity, but particularly in the thermal extremes. So it appeared that individuals with higher aerobic scope generally had higher activity, and theoretically, they would have a greater likelihood of encountering passive gear. So then we wanted to have a look to see what the link was between physiology and behavior. And so Lauren Bailey did a fantastic PhD, but what she first needed to do is take individuals using a repeated measure approach and develop individual aerobic then categorized those individuals based on their performance across temperatures as either low, intermediate, or high performance. She then took those fish into a lab and she did a series of behavioral experiments on those individuals, including activity, boldness, approaching a novel object, and aggression with a mirror in the tank and seeing how they responded to themselves. And what she found was very interesting. All of the the ones with high physiological performance, except one or two, were in fact bold. Okay, the bold, intermediate physiological performers and one bold, low performer grouped together in a cluster analysis, and the timid, intermediate physiological performers and subgroups of high and low performers grouped into a third cluster. So we then separated those out into those groups, and we did duels. And the duel comprised putting the fish in a tank at a certain temperature, at a range of temperatures. So we did 10, 18, and 21. And we took one bold, for example, a bold high performer, and bold versus a bold intermediate performer, timid high performer versus a timid low performer. And we did all the different potential opportunities, and then we plotted the results as a probability of winning. And you will see it was fascinating because at the lowest temperature, the timid low performers and bold low performers tended to win, okay? But they were dismal at 16 and 21 degrees. The bold high performers and bold intermediate performers won the duels almost exclusively at the high temperatures. So that really suggested to us that the probability of winning a duel at optimal and high temperatures was actually related to their physiological performance and not to their boldness, which says that there's an underlying physiological mechanism driving this whole thing, which basically is talking to our hypothesis that physiology might be the basic thing that is driving the response in these fishes. Okay, so to summarize, okay, first mechanism, we think that angling selectively removes high thermal performers, and I'm talking about the mechanism of driving vulnerability in populations. We think angling selectively removes high thermal performers because they're more active, firstly, and more likely to encounter gear across all temperatures. Okay, so firstly, they've got a higher aerobic scope across a range of temperatures, so they are more active all the time. They're more likely to encounter a hook and line. High performers are almost always bold, and they will get to the bait first at any temperature and win duels at the optimal and high temperatures. And timid and bold low performers may win a duel at cold temperatures, but because they're not active during those times, they might not even be competing. Okay. So to put that into pictures, let's imagine we have a high thermal performance individuals, high performance individuals, low thermal performance individuals in the population. We have fishing that happens across temperatures. At the low temperatures, the high thermal performers swim around more, they're more likely to encounter a gear, but if they encounter a gear at the same time as a low thermal performer that happens to be bold, it will take the bait first. But overall, the high thermal performers will outcompete them at low temperatures. At average temperatures, the low thermal performers might be around, but they will lose out in the boldness because they're not as bold. And then finally, at high temperatures, and this is interesting because of the climate impacts that we're having, it's the high thermal performers are the ones that are going to be caught out. And ultimately, if you have that selective pressure, you're going to end up with low thermal performers in your population. But what does that mean for the next generation of fish? So we went into the lab and we took fish from an exploited area and an unexploited area. We spawned them, we took them back to the lab, and then we compared their physiology. We couldn't use temperature because they are so thermally sensitive, and we were struggling to keep them alive, so we used pH as a stressor. And then we compared fish from an exploited area and an unexploited area. And we had a look, and we've got the control here, sitting over here, and fish from the exploited area, you'd see, went into a metabolic depression here of preflection. And preflection is a highly energetically expensive period where they move off from a yolk sac and are required to feed, and they sort of went into this metabolic depression phase. But then interestingly recovered quite well. But all the fish towards the end of the experiment from the exploited exploited population ended up dying. But with the the protected population, they survived through, and we had them for 6 to 8 months in the lab without any problem. And there was no metabolic depression in these early life stages. And we think that these were able to recover from the metabolic depression because we had a huge amount of food in the system to try and keep them alive. But if there's any lack of food in the system, I suspect that they may not have survived the stress. And so that really said to us that those genes, those high-performance genes, are actually being passed on to the next generation. And we suspect that this is fisheries-induced evolution, pushing angling vulnerability and actually influencing the way that fish are vulnerable to exploitation. So, okay, to summarize, how do we increase the vulnerability? How do we decrease the vulnerability? How do we decrease the vulnerability of fish angling? Okay, we need to protect our physiological diversity, first of all. Protected areas, super important. Keep those bold and physiologically fit fish around. Consider selective breeding programs where you use fish from those protected areas to, for example, stock the Murray cod. And then think about developing larval dispersal models. And this is marine, in the case of marine protected areas. So that the MPAs are placed in areas that seed the highly fished areas, so that we can keep our fisheries going. That's another opportunity. And then I'm very glad to see so many closures at warm temperatures in fisheries, voluntary closures, etc. That would really help. Closing fisheries at warm temperatures where those high physiological performers would be active and would be feeding would be a way to try and manage for these bolder and high performance fishes. So just some of our thoughts from some of the work that I did that we've been doing over the last little while.